CMorales, Bio 225, May 14, 2018, Accepting Technique and Culturing Microbes. I wash my hands thoroughly with soap and warm water. I put on my safety gloves, my face mask, my apron, and my goggles. I've disinfected my work surface by wiping the area with a 10% bleach solution, and I've also carefully poured some undiluted bleach into a bucket for my waste disposal, and anything that is exposed to microorganisms will also be needed to be treated in my undiluted bleach before I dispose of it. And I've also used my permanent marker to label one of the small plastic cups as alcohol. I've also placed my pipette in my cup of alcohol, making sure that the cup of alcohol enters the stem of the pipette. And I've also placed my candle on my work surface, and I'm now going to ignite the wick using my lighter. Now, I've also used my permanent marker to label my E. coli, my S. epidermis, and my S. cerevisiae. And I've labeled all those three on my nutrient broth. Now, I'm going to sterilize the lip of my E. coli nutrient broth with my flame. I'm going to grab 0.25 milliliters of the nutrient broth after I sterilize my E. coli. I'm going to be very careful when I grab this. And put this directly into my E. coli. I'm going to put the cap on it and shake it until my tablet has dissolved. I'm going to use a little bit more solution. And you're shaking it so that the tablet dissolves. It's almost there. However, I am going to add a little bit more solution just to speed up the process. Now the tablet is also dissolved. Now I'm going to use pick up all the solution and put it into my E. coli nutrient broth. Now I'm going to sterilize the edge of my E. coli, and then I'm going to close it. I'm going to repeat this step now for my S. epidermis. Again, I want to sterilize the edge. And also sterilize the S. epidermis right here. And then I'm going to go ahead and get 0.25 milliliters of my S. epidermis nutrient broth. And put it into this. I'm going to shake it till it dissolves. Almost there. It's all dissolved. Now I'm going to place this in my nutrient broth. Well, I'm going to add a little bit more. There's a little bit more remaining. Now before I close this one, I'm also going to sterilize it once more. And I close the cap. For the last one, I'm going to use the S. cerevisiae, and for this one, I'm going to put my yeast solution. So once again, sterilizing the lip of the tube. And this is my yeast solution. I'm going to grab 0.25 milliliters. And then I'm going to inject that into the cerevisiae. And before I close that, I'm going to again sterilize it. And then put the lid. Now for my cleanup, I'm going to extinguish my candle. And I'm going to put my 2 empty culture vials and my yeast container into my bucket of undiluted bleach for 30 minutes. And I'm also going to disinfect my pipettes that I have used with alcohol, and I'm going to pour my remaining liquids down the sink. And I'm also going to wipe my work area with the 10% bleach solution. I'm going to incubate my nutrient broth cultures for 48 hours. After 48 hours, I'm going to inspect my cultures for microbe. Developed cultures will either appear turbid or exhibit flocculent growth at the bottom of the tube. If cultures show no sign of growth after 48 hours, I'm going to keep them incubate for an additional 24 hours. Christina Morales, Bio 225, May 17, 2018. Aseptic technique and culturing microbes results from 60 hours of incubation time. Starting with the E. coli, the liquid is clear and there's a white substance that is formed at the base of the vial. With the S. epidermis, the liquid has turned cloudy and there is a very thin white substance that formed at the base of the vial. And then with the S. cerevisiae, the liquid is partially cloudy and there is a little bit of a thicker layer at the bottom of the base of the vial. Christina Morales, Bio 225, May 17, 2018, Asepting Technique and Culturing Microbes. I have my inoculation loops disinfecting in my alcohol cups. I'm going to be removing my microbe tubes that have been in my incubation area for 60 hours and transferring them into my agar plates by spreading them into each quadrant. So first I'm going to start with E. coli. I'm going to be sterilizing the lip. I'm going to very carefully enter my inoculation loop. And I'm going to spread it in the first quadrant of my E. coli. Before I close it, I'm going to sterilize the loop again. And then close it. Now I'm going to, using the same microbe that I already placed in the agar plate, I'm going to drag that on to the next quadrant. And sterilize. Then I'm going to go ahead and do the third quadrant of the E. coli. And sterilize my loop. Now I'm going to do the last quadrant of the E. coli. And spread it on the fourth quadrant. I'm finished with my E. coli, so now I'm going to move on to my cerevisiae. Again, I'm going to sterilize the loop. And I'm going to dump my alcohol into the first quadrant. I'm going to dunk it into the microbe. Remove it and place it into the first quadrant. I'm covering it up and then sterilizing the lid before I put the lid on. Now I'm going to repeat the process and keep moving the same microbe to the next quadrant. Now I'm going to move it to the third quadrant using the same microbe. And then to the fourth. And closing it. Now we are finished with the cerevisiae and we're going to move on to the epidermis. Once again, we're going to sterilize the lip. And then dip into the microbe. And transferring it into the first quadrant. Covering up the agar plate. And then sterilizing the loop before we close the lid. Now once more we're going to put the microbe into the second quadrant. And then sterilize the inoculation loop. Now we're going to move the existing microbe into the third quadrant. And close the agar plate. And then one more time. We're going to move the microbe into the fourth quadrant now. And close the epidermis. So my 3 agar plates are now going to be placed in my incubation room, where they will remain there for 48 hours. Christina Morales, Bio 225, May 19th, 2018. These are my developed colonies on my agar plates that have been in incubation for 48 hours. Starting with my S. epidermis, the shape appears to be rhizoid. The margins are entire. The elevation is slightly raised, and the size is punctiform. With my S. cerevisiae, the shape is circular, the margin is curled, the elevation is convex, and the size is small. For my E. coli, it's harder to visualize, but it is there. The shape seems to be irregular. The margin is lobate. The elevation is flat. And the size is pinpoint. Again, it's a little harder to see. But these are my 3 agar plates.